Okay, welcome to the last part of lectures 9 and 10. This is part 4. We're going to talk about the pancreas today, and we're also going to talk about pancreatic control. So let's get started. The pancreas has both exocrine as well as endocrine functions. In terms of its exocrine functions, it's one of the major organs that secretes digestive enzymes that will continue the breakdown of food that was begun in the stomach. In terms of its endocrine function, it mainly secretes insulin and glucagon that controls blood sugar. And we're going to talk about that in connection with the endocrine lectures. Now, one of the important jobs of the pancreas is to complete the digestion of proteins that was begun in the stomach. If you remember, the main thing that the stomach does digest is proteins because of the pepsin and the hydrochloric acid that's in the stomach. The pancreas is divided into two major sections. The head, which is the rounded portion that you see right here, is nestled in the C of the duodenum, which is the first portion of the small intestine, and the tail is the back portion here. As you'll notice, there's a main duct that runs through the tail, and as it gets into the head, it splits into two other ducts. One of these is known as the main pancreatic duct, and the one that's up here is known as the accessory pancreatic duct. Now, not everyone has an accessory pancreatic duct. One of the good ways to tell the main from the accessory pancreatic duct is that the main duct merges with the common bile duct, which is shown here in green. And these both empty together into the small intestine. The points at which they empty into the small intestine, you'll see the main pancreatic duct empties at the main duodenal papilla. The accessory pancreatic duct empties at the minor duodenal papilla. Now, if you look on the lower right of this slide, you'll see a blow-up of where the main pancreatic duct and the common bile duct come together and then empty into the small intestine. So here we have the small intestine itself. And you notice that the common bile duct, which is here, and the main pancreatic duct come together, and they empty into the small intestine at a junction called the hepatopancreatic ampulla. Now, both of these ducts, before they enter the small intestine, are surrounded by a ring of smooth muscle that's known as the hepatopancreatic sphincter. Between meals, this ring of smooth muscle is contracted and keeps bile and pancreatic enzymes from entering the small intestine when there's nothing in there. After we eat, there are a set of circumstances, as we'll see in a couple of minutes, that will actually relax this smooth muscle and allow both bile and pancreatic secretions into the small intestine to begin digesting the food. In this slide, you see the blood supply and the drainage of the pancreas. So the arterial blood supply of the pancreas, you'll see, comes from a number of different sources. Gastroduodenal artery, the superior mesenteric, the superior pancreatoduodenal artery, the splenic artery, as well as smaller pancreatics that all feed the pancreas. So here we see, of course, the head, and here we see the tail, as we just talked about. Now, the venous drainage of the pancreas is accomplished by several vessels: the pancreatoduodenal vein, the splenic vein, as well as the smaller pancreatic veins. As we mentioned previously, all abdominal organs drain first into a common hepatic portal vein before draining back into the major circulation of the body. And that's the case with the pancreatic drainage as well. You'll notice that all of these veins that drain the pancreas merge and enter into the hepatic portal vein. This goes into the liver, divides into capillaries, and eventually this will form the hepatic veins that will drain into the inferior vena cava, which we see right here. Now, in terms of the things that the pancreas actually secretes, we call this pancreatic juice, and it secretes a number of different enzymes as well as some other substances that are important to its function. You'll notice that the pancreas secretes a variety of different enzymes. Amylase, which we talked about previously when we talked about salivary amylase, as splitting large carbohydrates into simpler ones. So this will split, for example, glycogen into disaccharides or smaller units. Lipases break down triglycerides. Nucleases that break down nucleic acids such as DNA and RNA. And in addition, the pancreas also secretes proteolytic enzymes. You'll remember that proteolytic means protein digesting enzymes. We're going to talk about this in a second. I want to talk first about the bicarbonate ions that are secreted by the pancreatic duct cells. The cells that line the main pancreatic duct secrete bicarbonate ions. These are very important because what they do is to make the pancreatic juice alkaline at a pH of about 8, and so neutralize the acid that comes from the stomach. Remember, the chyme that comes from the stomach and goes into the duodenum, the first portion of the small intestine, is acidic, and one of the things the duodenum doesn't have that the stomach has is a thick layer of mucus coating it so that it can be protected from acids. So it's very important that the acids coming from the stomach be neutralized very quickly to protect the wall of the duodenum. And this is accomplished by bicarbonate ions and several other basic ions that are present in pancreatic juice and raise the pH in the intestine to around 8. One of the other things you'll notice is that pancreatic enzymes all function at an alkaline pH. If you remember, pepsin that we looked at in the stomach functioned at an acidic pH of around 2 to 3. The enzymes that we have present in the intestine function, though, at an alkaline pH. Let's take a look at the pancreatic proteolytic enzymes. Before we do that, realize that if the pancreas secreted active proteolytic enzymes, that is, protein digesting enzymes in their active form, these might very well start digesting the pancreas while the enzymes are present there, and so it might actually harm the pancreas. So the strategy that the pancreas has for doing this is secreting these enzymes in inactive form. And we see a few of those here. In fact, these are the main enzymes that the pancreas secretes. We see trypsinogen, chymotrypsinogen, and if you remember, the OGEN on the end of this means that these are in their inactive form. We also have an enzyme called procarboxypeptidase, one called proelastase. All of these are what are called proenzymes or zymogens, meaning that these are in their inactive form. That begs the question, if these are in inactive form, when do they get activated and how do they get activated? Well, they don't actually get activated until they reach the lumen of the small intestine. And how that happens is because the brush border cells of the small intestine, the cells that are lined with microvilli, secrete an enzyme known as enteropeptidase. Sometimes this is called enterokinase. And the job of this enzyme is to convert trypsinogen into its active form trypsin. And that's a very important key conversion because trypsin will convert all these other enzymes into their active forms. So all of these enzymes now are the active forms of the inactive enzymes that were secreted by the pancreas. You see chymotrypsin, carboxypeptidase, and elastase. And the job of all these enzymes, of course, is to take the proteins that are in the small intestine that are partially broken down from stomach digestion and complete their digestion. And you'll notice that this is going to break the proteins down into smaller units like dipeptides, tripeptides, and in some cases, amino acids. So as we said before, the purpose of the proteolytic enzymes secreted by the pancreas is to continue the breakdown of proteins that began in the stomach. Now let's talk a little bit about the regulation of pancreatic secretions. We don't want a continual flow of enzymes from the pancreatic cells through the pancreatic ducts. One of the reasons for this is this ring of smooth muscle that I mentioned before, which is the hepatopancreatic sphincter, is tightly closed between meals. Now, if these inactive enzymes were to be secreted and started to back up right here because they couldn't get out, they might very well become active in here. If they stay static and stagnant, there are things that might activate those enzymes. That's not a good situation to have. So what we want is we want to downregulate production of enzymes and secretion of enzymes by pancreatic cells, except for after meals. If you look on the top of this diagram, you'll see an acidic bolus of chyme coming from the stomach through the pylorus and into the small intestine. This is actually an activation signal for the pancreas to begin secreting enzymes and also for the hepatopancreatic sphincter to relax. Let's see how that works. First of all, when acidic chyme comes into the small intestine, we have secretion of something called CCK. This is called cholecystokinin. I'll spell it out for you. Cholecystokinin. This is actually a hormone. So cholecystokinin will go into the blood, will come back, and inside the pancreas, what this will do is it will upregulate production as well as secretion of pancreatic enzymes and all those zymogens that we talked about before. In addition, when this acidic chyme goes into the small intestine, we also activate the parasympathetic nervous system. This also upregulates secretion of pancreatic enzymes. Now, one other thing the acidity of the chyme that's coming from the stomach does is it causes secretion of something called secretin. Secretin is a hormone that's secreted by the wall of the small intestine, goes into the blood, comes back around to the pancreatic duct cells, and it stimulates them to release a watery pancreatic juice containing bicarbonate, as we talked about before, as well as phosphate buffers. Both of these will serve to neutralize the acidity of the chyme that comes into the small intestine. So secretin is another important hormone secreted by the wall of the small intestine, as is CCK. We'll see that CCK has also a number of other functions as well. So we can say that this is more or less a feedback loop that we have here. And as you can see in this diagram, starting at number 1, the acidic chyme enters the duodenum. The intestinal mucosa releases secretin, also causes the release of CCK. This causes the pancreatic cells to start releasing a watery type secretion, pancreatic juice, that contains bicarbonate ions and phosphate ions, and these in turn will serve to neutralize the acids that are in the acidic chyme coming into the small intestine. Now, this is another chart similar to the one that we saw with the stomach. I know once again this looks kind of busy, but this is really an important overview of some of the functions of the pancreas and how the pancreas is controlled. It also has some additional stuff on it that will serve you well once we start talking about how the intestine works as far as digestion and absorption. So this might be a good figure to keep handy when you're looking at the individual parts of digestion and how the pancreas, how the gallbladder works, and how the intestines actually process food and where the digested food actually goes. Let's take a look at this. First of all, we start with this event, right? The acidic chyme comes from the pylorus and enters the duodenum. As we said a couple of minutes ago, that's a major signal. One of the signals that we talked about before, and you'll notice this is a stimulatory arrow, as you see in the key up here in the upper right. So when chyme enters the duodenum, it causes secretin to be secreted into the blood. This is a hormone. This comes back to the bile and pancreatic ducts, causes the release of bicarbonate and phosphate buffers, and this is going to raise the intestinal pH to about 8. This not only neutralizes the acid in the chyme, but it also provides the most favorable environment for all the enzymes that are present in the small intestine to work. So this is one thing that happens as a result of chyme entering the small intestine. The second thing that happens is when chyme comes into the small intestine, as we said before, there's a very important hormone controlling digestion that's called cholecystokinin. And the cholecystokinin, or CCK, does a number of things. One of the things we already talked about was the fact that it activates the pancreas to secrete all its enzymes. So let's take a look at how this works. You'll notice that cholecystokinin causes the pancreas to upregulate its secretion of enzymes. We're talking about enzymes like lipases that digest fat, nucleases that digest nucleic acids, amylase that will begin digestion of complex carbohydrates, and finally enzymes to digest proteins. But if you remember, as we talked about before, the pancreas is going to secrete these enzymes that digest proteins in inactive form first. And we mentioned these inactive forms of the enzymes right here that we call proenzymes or zymogens. What's going to activate this whole sequence, remember, is something that's secreted by the small intestine itself. This was known as enterokinase or enteropeptidase. This is going to convert trypsinogen into trypsin. Trypsin in turn is going to convert all of these inactive forms into their active proteolytic forms. This is going to begin digestion of proteins, and now we're going to start creating di and tripeptides. Eventually, some of the enzymes that are present in the small intestine itself are going to convert these into amino acids, so into the basic building blocks. The amylase is going to convert glycogen and starches into mono-di-trisaccharides. Some of the small intestine enzymes are also going to convert these into smaller units, the monosaccharides. And then finally we have nucleases. The nucleases are going to digest nucleotides, and these are going to all be absorbed into the portal vein. I want you to notice this because this is important. Sugars get absorbed into the portal vein. Amino acids get absorbed into the portal vein. Nucleotides get absorbed into the portal vein. There's one substance that's a result of digestion that does not make it into the portal vein, and that is fats and fatty acids. These fats and fatty acids are instead absorbed by what we call lacteals, which we mentioned earlier. These are in the villi of the small intestine. These were lymphatic capillaries. Okay, so just keep that in mind for now. We're going to come back to this a little bit later. Now, there are a couple of other things that cholecystokinin does that are important. We know that it activates the pancreas to secrete enzymes, as we just saw. The other couple of things that it does is, number one, it causes relaxation of the smooth muscle in the hepatopancreatic sphincter. There would be no sense in the pancreas upregulating its secretion of enzymes if they couldn't get out of the pancreas into the small intestine. So these two things are coupled, secretion of the enzymes as well as relaxation of the hepatopancreatic sphincter. And this allows the enzymes that the pancreas does secrete to get into the lumen of the small intestine. Now, one other thing cholecystokinin does, as we'll talk about when we talk about the gallbladder, is it also causes the gallbladder to contract. Inside the gallbladder, we find very concentrated bile, and the bile is very important to aid in the digestion of fats, as we'll see when we talk about this in the next couple of lectures. So this is, once again, by way of overview of how the pancreas functions. I think this slide is very important, and I really do think it's important for you to keep this alongside the other figures when you're studying the individual pieces of this to see once again where they fit and how they work. The same was true of that large chart that we looked at as far as stomach control. So it's important that you try to get a big picture of what's going on here and then study the individual parts. I think this will all start to make some more sense to you. Okay, that'll finish up part 4 of lectures 9 and 10. I will see you for part 1 of lecture 11 next.